Hi, everyone. So my name is Matt Disney. I'm a professor in the Department of Chemistry at Scripps Research. Today, I'm going to tell you as part of our Front Row series about work that I've been doing in my lab on trying to develop a framework to understand how to make medicines for diseases that are very difficult, if not impossible to treat, that include cancers that have a poor prognosis, the most common adult onset form of muscular dystrophy. And also, I'm going to tell you about some recent work that we've been doing on trying to tackle emergent pathogens such as coronavirus. And so as many of you know, Scripps Research is, we're a research institute. That research institute is built on research groups, and the foundation of those research groups are the people in them. And so although I'm a representative of about 20 to 25 people that are in my lab as any function of time, our group comprises people from all nationalities, from all over the world, people from Japan, China, India, etc. And also people at all level of training. We have graduate students in the lab. Here are a few of the graduate students in my laser pointer: Ali Angibello, Samantha Meyer, Jessica Bush. We also have postdoctoral fellows and staff scientists. One thing how I start off most of my talks is I thank people that provide us financial resources, and most of that comes from the US taxpayer. So we get a wide variety. The vast majority of our funding is appropriated and delivered by things like the National Institutes of Health. The reason that happens is a lot of what we do, as you'll see today, is basic science. And so by basic science, I mean that we study things that probably initially wouldn't have an application, but the thought is that we would set a scientific foundation by which broad applications can be derived from. Now, probably my group is more applied than most people that are in the basic science area, so the applications that we have are going to be more apparent, but it's basic science nonetheless. So we have a wide variety of funding by both the NIH. Part of my lab is a neurological lab, so we have funding from the National Institutes of Neurological Diseases and Stroke. And we also get philanthropy funding that we're blessed to have from the Nelson family, Susan and Alan First, and the Women for Breast Cancer Research at Frenchman's Creek. One thing I want to tell you about the students that we have at Scripps is in my travels as a scientist, I go not only to a lot of universities, the top universities in the world, but also a large number of pharmaceutical companies. And so always and inevitably, a person that runs a large drug development program at Pfizer or a large one at Merck are people that have been trained at Scripps. And so I think the faculty here are really blessed to be able to work with young people that we're going to have a hand in training. And their impact is going to go well beyond what we're doing in the laboratory. That's their training ground, but they're being trained for tackling significant and serious diseases that afflict patients for decades after they come from the lab. And so what does my lab do in sort of one sentence, and why do we do it? And so we study the basic science of how RNA folds. So I'll tell you what RNA is in this talk. And by understanding how RNA folds, we also try to understand how RNA folds in a manner that it'll bind to small molecules. And so small molecules are pills like aspirin that we would take to treat, if you have, a headache. Could also be things like Lipitor if you had high cholesterol. And the reason that we study how RNA folds and binds to small molecules is we want to use that basic science to give us new insights into how to develop new approaches to treat diseases. And so schematically, what my lab really does is we try to understand areas of medicine. So we try to understand what are genes that cause disease, what are the implications of when those genes are mutated to trigger a cascade that causes serious disease. So those diseases that I'll talk about today are cancer, muscular dystrophy, and also coronavirus. Things that we also work on are studying how genes cause things like ALS. And by studying the way that those genes function and integrate in a host, we can hopefully understand how that gene integration can utilize and identify new therapeutic targets. And one thing that my lab is centrally focused on is trying to understand how the RNAs that are produced under these disease settings can cause disease. And then we like to deliver lead small molecule medicines that target those diseases very quickly and very rapidly. And so really we look at a patient to identify genes that cause a disease, and then we try to deliver a medicine or a small molecule we're going to talk about, which is basically a medicine in a pill that can perturb that gene function. So right now we're studying perturbation of genes, but that perturbation with that small molecule can be evolved into a lead medicine. And so I talked about my lab and my scientific career has been focused on studying RNA. And so RNA is very similar to DNA. Most of you are well aware of the central dogma of biology, where our genes that were given from our parents. My parents were blessed with 7 children, so they gave those genes many times over. That genetic material that you inherit from your mom and your dad gets decoded into RNA. And so your RNA is basically, was thought of in the 70s as an intermediary between DNA and protein, and that RNA provides the instructions on how to make protein. Now we know very differently. And so again, my lab is focused on studying RNA, which is the central portion of this slide. And my scientific life is going to be devoted to that because I think it's so important. The reason that I think it's important is that as people, and I'll go into this, started to sequence the human genome, they started to realize that RNA was playing a very important role in terms of being a medicine target, but didn't know how to target it. And so my group has really been trying to push forward new approaches to drug RNAs with small molecules. And so we think about developing therapeutics. People think about DNA. And so DNA is a well-known therapeutic. Actually, some of the first in line cancer therapeutics that are used to treat patients like that have cancer are molecules that just target and damage DNA in a patient. When drug discoverers think about RNA, they have no idea about how to drug it, but they know it's important. And most of modern drug discovery, which I'll tell you why, today has been focused on proteins. And so a drug discoverer today that studies small molecule medicines to treat human diseases, they kind of know how to target a protein. And so the reason that they kind of know how to target a protein is focused on how a protein folds. And so proteins are made of diverse building blocks that have, and because the individual repeating building blocks or bricks that are used to build proteins are very diverse, then the way that one can construct a fully functioning protein out of those diverse building blocks can be very complicated, right? And they're obviously the dominating target of small molecule medicines. And so, for example, many proteins can look like water and look like oil. Many of them can, have a charge like a battery, etc. And so in modern medicine, and there's 20 different natural amino acids that are used to build proteins. And so in modern medicines, proteins are the predominant target of those medicines. And so here are 3 examples. From left to right, Gleevec, which is a molecule that's one of the first early precision medicines that was developed for cancer patients that could target a specific gene that was causing a cancer. It targets a protein that is upregulated massively in cancer. Also, drugs that target heart disease can be bound by small molecules, and also obesity-related targets. And what you can see within these folds is that they're very compact, they're very complicated. In fact, they're so densely packed that in order for you to be able to see the binding sites for these proteins, the drugs in these proteins, I had to peel away parts of that protein so you can see it, right? They're very densely packed. And it's densely packed in such a way where there's a unique interaction between a protein and a small molecule drug that targets it, much like a key will go in and only open one lock. So there's very significant specificity that proteins have when they target when they're targeted by small molecule medicines. And so we're living in an era of modern medicine that's, I think, very exciting. I'm very jealous of my nieces and nephews and my children because the amount of information that we have now is just amazing. One example of that is the pace at which we can acquire human genome sequence. And so in 2001, when President Clinton was in the office, many of you can remember this, Francis Collins and Craig Ventnor were part of a White House celebration where in 2001, they published the first draft of the human genome. From that time, the first human genome sequence cost about $100 billion. But rapidly in the past 15 or 16 years, we've been able to get to a position where we can sequence a genome for about $1,000. And I think that's absolutely amazing. And to put that into context, what's shown here in this orange line is what's called Moore's Law. And so Moore's Law says that you can double the computational power that you have on a computer every year. And so the consequence of Moore's Law is that the computational power that got a man on the moon, right? We just celebrated the 50-year anniversary of getting a man on the moon, but that computational power to get a man on the moon is about 100,000 times less than the power that we have sitting in our pocket with our iPhone or our iWatch. And I find that completely amazing that the efficiency that we're able to acquire genome sequence is far outpacing that. Imagine 50 years from today, how rapidly we'll be able to get genome sequence or do other aspects of scientific exploration. Now, one of the first consequences of the human genome was that we had about 1/3 of the genes, humans had many less genes than we thought. And one of the other things that people started to think about was once we had this massive amount of genome sequencing, how could we use it to enable the development of therapeutics? And so one thing that people first did is they realized, and I mentioned this earlier, is that only about 1% of our genome encodes for protein. And so in that early central dogma slide, I showed that protein was the input of biology that was thought about in the 70s. And so in early 2001, it was identified that only about 1% of our genome was converted into protein. And in fact, 80% of our genome was converted into RNA. And so there are many consequences for that. So for example, one of the consequences that if we want to understand the differences between us and when I grew up, my best friend was my dog. If I want to understand the differences between me and my dog, you wouldn't want to look at our protein coding genes because they're not as different. They're not as different as, for example, our RNA that we're making. Actually, the difference between species as a function of complexity is most closely tracked to the differences in the RNAs that we produce, not the small fraction of our genome that gets converted into protein. So not only can differences in species be associated with differences in complexity of RNA, but it turns out there's basically every disease that affects man, either a cancer or ALS or muscular dystrophy, but also coronavirus, can be caused by a malfunctioning or a disease-causing RNA. And so the consequence when people first looked at druggability and genome sequencing is they realized that, as I mentioned earlier, people knew how to drug protein. And it turns out that about 15% of protein families have been drugged with a small molecule medicine or targeted with a lead small molecule. But one of the intriguing possibilities for me, given that I was trained as an RNA scientist, is that actually zero, for all intents and purposes, zero human RNAs have ever been drugged with a small molecule. And this turns out to be important because when one goes to look at the associations that RNA has with diseases, it's nearly every disease, as I mentioned earlier. And so this was on October 15th of 2019, I just did a PubMed search. So I did a search of the published literature, and the amount of publications for RNA that is associated with the disease is rising exponentially. And many of the diseases that come up include things that are very important for many humans that walk the earth are afflicted with these diseases, such as cancer, diabetes, stroke, dementia. My mom had Alzheimer's disease, and so I'd really like to understand, she died of Alzheimer's disease. I'd really like to understand what's the role of RNAs in dementia, to figure out how I could treat somebody that was going through what mom was dealing with. Fibrosis, so if you smoke cigarettes, you can have damage to your lung that can be COPD, but it can also be fibrosis. And it turns out that there's many RNAs that are associated with fibrotic damage, lung damage. And also in 2011, I'm not going to have time to talk about this, but in 2011, it was found that the most common cause of ALS was a toxic RNA that families actually inherited from each other. And you could basically trace whether or not someone had ALS that they were inherited by having this long toxic RNA. And we have a huge program in lab on learning how to treat, learning how to drug these ALS associated diseases. However, when we started doing this about 15 years ago, you know, we told the scientific community, because we had to get research dollars, and the way you get research dollars to run a research program is you have to apply to NIH. And in applying to NIH, you have to get your colleagues to review your grant and say, hey, you know, I think this is worthy of investment. And many people, when we started to do that, thought, hey, Disney, you're crazy. This is never going to work. And the reason that they suggested, or they thought pretty aggressively, that drugging RNA was a waste of time or never going to work goes back to the building blocks that RNA is built from. So RNA is only built from 4 building blocks. So these building blocks are called purines or pyrimidines. And so those building blocks are only assembled, or it was thought to only be assembled in 2 ways, namely base pairs. And that is much less diversity than the 20 building blocks that were built with proteins. And so many people thought, hey, you know, if you're going to make a medicine that targets an RNA, RNA has to fold into a structure that that medicine can bind. And RNA is only built from 4 different things. If it's only built from 4 different things, it's not going to fold in a very unique way that you can get a medicine to bind to it. And so in some sense, RNA looks like El Capitan, right? This big sheet of rock that's emerging from the earth that doesn't really have a lot of cracks and crevices that you can hold on to it. Because as I showed earlier, the way that medicines bind to protein targets is there has to be a shape, or a defined shape, that you can actually stick into it and hold on to it to inhibit and affect its function. And so just like people like Alex Harnold have developed creative and very brave, to put it mildly, approaches to scale things like El Capitan, we think we've developed ways in which you can identify quickly what are the types of RNAs that'll fold into pockets, and then develop creative ways to affect their function, to put forward RNA as being not at an afterthought of medicine and developing drugs, but really at the forefront. Because if we can bring RNA to the forefront of medicine development, it's going to unlock a lot of potential to drug nearly every disease, and many of which drug companies have worked on for decades, and there's just not really a protein that you can go after to affect the disease in a very efficient or effective way. And so one thing that we know, actually I got my PhD studying how RNA folds, is that we know RNA is a single-stranded biopolymer, and in contrast to what people think about in terms of RNA folding, despite being comprised of 4 bases, is that RNA can actually fold in very complicated ways. And so here's some way that RNA folds, is shown here. Here are RNAs that play key roles in very important diseases. So this is an RNA that's associated with dementia. Here's an RNA that's associated with Parkinson's disease. This is an RNA that's associated with cancer. Here's an RNA associated with heart disease. And here's that RNA that I talked about earlier that causes the main genetic cause of ALS. And so what we've been able to do, and I'll show you how we've done this in a very basic sense, is been able to identify the pockets within these RNAs that fold, and have been able to deliver medicines that can bind them very tightly and very selectively to affect the biology in a manner that would be in a therapeutic benefit. And so when we started getting into this area, because it was very controversial, it wasn't known if RNA would bind to medicines very tightly and very selectively. And so we basically started our journey that we've been on for the past 15 years by asking a very fundamental and basic science question. And so that was, 1, does RNA fold in a manner that allows it to bind medicines? Number 1. And the second question is, what are the structures and shapes of the medicines that bind RNAs? Because if we thought if we could identify this basic science question, we would unlock a key to drugging all these RNAs in a manner that would allow us to do it by just looking at genome sequence. And I showed you earlier that we have a lot of that. And so the way that we go about doing this is we take a small molecule or a medicine that you're going to take in a pill that's shown here in this yellow sphere, and we basically ask the small molecule, we go, hey, if you're given hundreds of thousands of combinations of RNAs, can you tell us what are the RNA structures you like to bind? And so we do what's called a massively parallel selection, which allows us to define an RNA three-dimensional fold that binds to this drug. And so my group has been working on this platform for 15 years, and we've identified a huge amount of information on the types of RNAs that fold medicines, right? And we do this in a manner that is completely disassociated from a disease. So we're just asking a fundamental question and saying, hey, what are the RNAs that fold and what are the medicines that bind them? And so the reason that we did that is we thought that there was such a large encyclopedia of genome sequence out there that if we had a lot of this information, we could go and apply this information across disease indications to identify small molecules that could perturb in a very precise disease-associated pathway these RNA targets to have a therapeutic benefit. And so the way we went about doing that is not in a manner that's so dissimilar for using an Enigma machine. So an Enigma machine is shown here. This is the machine that people like Alan Turing, who was a computational scientist from Britain, and he developed a way where he could decode messages that the German soldiers were sending to each other in a very significant way by using this Enigma machine, which basically had an algorithm, and it could take what the Germans were sending and decoding it so that the British, the Allies could understand what was going on. And this is one significant advantage that we had to winning World War II. And so we wanted to develop what's called a code breaker by using this information on RNA folds that bind small molecules. And we call this approach Inforna. So I'm going to tell you how we do this. So it's basically information about RNA. We've merged those two words to call Inforna. And so what we have now is there's been a lot of work done on trying to understand basically just by using RNA sequence, what are the regions in an RNA that fold and what do those folds look like? So basically what we can do is we can take an entire human genome and we can fold an RNA. So I'm going to illustrate what that means by using this purple line. This purple line is to represent one RNA across 30,000 that your cells may produce. And so we can do a calculation by using a computer and we can look at that RNA. And that RNA has two regions of within it. It has one region that's not folded, that still has this squiggly line. And it has two regions that are very robustly folded. And so this is something that we call has a high probability of structure or is very likely to adopt a very compact three-dimensional fold. And when it adopts, again, that fold, it should be druggable with a small molecule. And so because we have this information on the RNA folds that bind small molecules, and we have the ability to look at the human genome and identify regions within RNA sequence that are robustly folded, we can ask ourselves by using a computer a very simple question. We can go, hey, this RNA is folded in this certain way. Do we have a small molecule that binds to that fold by using the experiment that I just showed about 3 slides ago? And so that's what we go, is this a druggable pocket? And so here's a 3-dimensional rendering of that fold that's shown here. And then we can identify, are there drug-like medicines that stick in these pockets, which is shown here in the yellow structure. And we can do this by using the entire human genome. This tells us, one, is it a druggable pocket? The other thing that this calculation does is it says, hey, if you bind to the RNA at this site, are you going to affect its function in a way that is beneficial for making a therapeutic? The other thing that allows us to do across the entire human genome is it can tell us, hey, you know, this is an RNA that causes cancer and you have a drug that binds it, but that RNA is also present in an RNA that makes all of your protein. And so you really don't want to use that molecule because it can cause toxic side effects. We can then ask ourselves another question: is there a second molecule that's more selective, that specifically targets the cancer-causing RNA and doesn't target these RNAs that are needed for healthy cells to live? And when we do that, we can very upfront quickly study and pretty rigorously define if the molecules are going to be very selective and specific for a given patient type of cell. Now, these molecules can bind RNAs to affect their function, but we've also been able to develop our approaches where we can trigger RNA, small molecules, to actually destroy a toxic RNA in a cell or an animal and rid that toxic RNA altogether. And we've been able to do this in one of two ways. One way is we can basically directly cleave an RNA target, which I'll show in this talk. The other way is we've been able to basically programmably awaken your immune system and get your immune system to be awakened to catalytically destroy an RNA. So one copy of that small molecule that targets an RNA can cleave up to 100 copies of a disease-causing target, and do this in a very precise and specific way. All right, and so again, this technology we've applied broadly, but I'm going to talk about applying the applications that we've had in the lab to what I would consider two very significant human diseases. And then in the end, I'm going to talk about work that we're doing against coronavirus. And so many of the diseases, I'm focused on diseases that I think I can make a largest impact in. And so I think one of the amazing things about having my job is I don't ever think I come to work in a day, and the work is never done. So, as my wife can tell you, I like to work a lot and think about the stuff we're doing in lab, because it's not a job to me. It's my vocation. It's what I think I need to be doing while I'm on the earth. And so the diseases that we think about are things like cancer, so cancers that are hard to treat, and also human genetic diseases that range from Fragile X syndrome, that's the most common single gene form of autism. We've worked extensively on Fragile X syndrome to not only study how the gene, it's actually a gene is turned off permanently in these children. We've identified in collaboration with Sami Jaffrey Group at Cornell Weill, the pathway by which this gene is turned off. And we've also developed approaches to turn it back on, which could have a therapeutic benefit. We've also worked in myotonic dystrophy, which I'll tell you about, ALS and dementia, which I'm not going to talk about due to time. And we also work in Huntington's disease. I wanted to take a second here and tell you that a person that I've worked with in my career is someone called Nancy Wexler. And Nancy Wexler is one of my science heroes, because her mom, unbeknownst to her, actually suffered from Huntington's disease before they knew what the gene was, what the gene was that caused the disease. And so Nancy, about 30 years ago, because her dad was Milton Wexler, and he funded the Hereditary Disease Foundation, and they got funding to try to understand what was going on with Nancy's mom. And Nancy, who's was a professor at Cornell, actually utilized the resources there to bring together scientists to try to identify how what was the gene that was affecting mom, right? And so she was able to identify a patient population with others in South America that had a very high incidence of Huntington's disease. And Nancy was able to work with these people to get biological samples, and scientists such as Jill Bates and David Hausman to really identify the position of that gene and how Huntington's disease is caused. And so, this was about 30 years ago. This was the second gene that was ever associated with a disease. And only about in the last year and a half did we have the first therapeutic that could lower Huntington. And so these things takes time, but without the foundation and motivation of people like Nancy, who knows where we'd be today? We certainly wouldn't be as far along in understanding the disease and elucidating new therapeutic approaches. And so there was a profile in the New York Times that was written last week on Nancy. Actually, she does have Huntington's disease that she disclosed. And I'm telling you, that's something that you can read about. It really shows you the journey of these patients' families to really enable scientists like me to understand basic biology and leverage it to make therapeutics. What I'm going to talk to you in the first module here is that breast cancer is one of the diseases that we've been going after, which cancer generally affects 1 in 20 people. My dad had lymphoma, and actually, when he had lymphoma, he was given rituximab, which is an antibody. It's a humanized antibody. And Dad was a very, very tough guy. And I'll never forget when he told me he had this. He was very emotional. It's probably the second time in my life I ever heard my dad get emotional. And then about a week later, after he started taking this humanized antibody, he called me on the phone. I'll never forget it. And he could actually feel that he was getting better. And so, you know, it's amazing what science can do. And people like Richard Lerner, who was our former president of Scripps, he developed the humanized antibody technology. And it was very interesting. Dad didn't know that Scripps played a role in this, but when he found out about that, he was even, I think, more proud about me being here. And so breast cancer affects 1 in 8 women. When women have breast cancer, the first thing that they try to do is they identify if this breast cancer is something called triple negative breast cancer or non-triple negative breast cancer. If a breast cancer is non-triple negative breast cancer, that means that we have targeted therapeutics that can be easily deployed to treat this cancer. Unfortunately, about 30% of breast cancers you can treat non-triple negative breast cancer. That means there are medicines that can be used to treat these genes. If you have triple negative breast cancer, there's no precision therapeutic that can be used to treat this cancer, although there was one that was approved in the past few months that can treat a segment of these patients. And so, so one question that we had by deploying our technology is, could we develop a lead chemical probe that could target an RNA that was associated with triple negative breast cancer to give some insights to see if one can target the RNAs associated with this disease as a precision medicine. And so we utilized our approach, long story short, where we knew the encyclopedia of RNAs that were produced in triple negative breast cancer patients. We identified one that was associated with cancer. So it actually suppresses death of cancer. One way that cancer survives is it stops a normal death process that cells undergo called apoptosis. And so we could identify 2 small molecules that could bind to 2 different folds in this RNA target. And so basically, long story short, is Inforna identified these 2 compounds that bound to 2 sites in an RNA, and we could link them together. These molecules would bind with very high potency and very high selectivity to this RNA target. And so when we deliver the 2 halves of these compounds, they have no effect on triple negative breast cancer. This is the fraction of cells that undergo programmed cell death. When we take this active medicine, which is called this dimer, and give it to non-disease affected cells, these cells have no effect. However, when we deliver this molecule at a very low dose to triple negative breast cancer cells that were cultured from a patient, 90% of those cells undergo this apoptosis. So we basically change their protein expression profile in a manner where the cells actually kill themselves. They eliminate themselves. They undergo this apoptosis that they normally don't go under. And this was a very rapid development of this compound. In our lab and at Scripps, we have to do studies in mice. This molecule, when a mouse has cancer, we can deliver it to them and it'll shrink the tumor burden in the mouse. That's what's shown here. This is just an image of a tumor in the mouse. This is when we give them our drug. And you can see that that tumor burden is greatly diminished in these mice. Another issue in breast cancer is that patients not only can die from the original cancer, is that they often have significant issues due to something called metastasis. And that is when you get breast cancer originally, people oftentimes don't die from the tumor that's in their breast. They have issues where the cancer grows into your brain or your lung. And when you get a metastasis or a second site of cancer within these patients, that's when things really start to get tricky, because the cancer is overgrowing in many other areas. And so one of the challenges that we had within Inforna is, could we develop molecules that can stop metastasis of breast cancer? And so many RNAs are upregulated and causative of metastatic cancer. And so we utilized Inforna to identify a molecule that could target an RNA that causes cancer metastasis very rapidly. And we could deliver this into a mouse lung where basically what's shown here to the left here is a lung that breast cancer is growing on it, because when you deliver these metastatic tumors into mice, you actually get breast cancer growing on the lung. And that's what's shown here in these dots that are on that lung. When we deliver this molecule, you can see that there's less of these nodules that are growing on the lung. In fact, they're significantly reduced. And in many cases, they're gone. And actually, in the way that this molecule works is it binds to this cancer-associated target, and it reawakens the immune system to where the cells will actually rid themselves of this target in a mouse and stop breast cancer metastasis. And so we're really trying to work on two very significant areas in cancer. One is targeting triple negative breast cancer, so to stop its growth before it becomes metastatic. But the other issue that we wanted to address within the cancer space is figuring out ways that we can target metastatic disease to stop it. Other work that we've been doing in the lab is how we can also take cancer cells that are not sensitive, or they're resistant, they're just not sensitive to known chemotherapeutics, and there are RNAs that are produced that are making these molecules, these cancer tumors be resistant to therapeutics, and you can actually make them become sensitive. So we can develop a precision medicine way where we can resensitize cancers to known therapeutics. And because we're doing this by RNA, we can take, in principle, you can take a tumor from a patient, you can sequence the genome and identify all the RNAs that are expressed in a cancer. And then one thing that we'd like to do going forward is figure out a way to massively scale this up to develop precision approaches where one day you can go to your pharmacist, and your pharmacist may read out from your tumor all of your RNA genetic composition for your cancer. And rather than give you one medicine, they can say, oh, there's 4 genes that are upregulated here in your cancer. We don't want to give you one medicine. We want to give you these 2 combinations. And patient A can take cancer, maybe takes drug 1 and 3, and patient B may take drug 1 and 16. And so we really want to get into an era where we can personalize treatments based on the genetic composition of these patients. Now, we're just starting to do that, but it gets excitingly provocative to think about how we can drive science to really get us there probably quicker than we would ever think. I mean, this is another way where I'm really jealous of my children and my nieces and nephews. I mean, they're more likely to see that day than I am. The other area that we work on are human rare diseases, so genetically defined rare diseases. So rare diseases are diseases that affect, 1 in 1,000 people. So, much of basically, in order to get the pharmaceutical companies to be interested in rare diseases, the FDA and the federal government has to make an economic incentive for them to do it, because there's just not as many patients that will be able to take their medicine. And so they have to be massively incentivized. I think one of the advantages that we have in the Research Institute is those diseases that are rare, oftentimes on aggregate, are not as rare, and we can sort of understand the basic science in a very robust way to really lay the foundation for a big drug company to come in and make a medicine for this disease later on. And so in some sense, we're de-risking their investment because we're building a solid scientific foundation from which a Merck or a Pfizer can come in and think, oh, the scientists have laid a foundation of basic science in this rare disease where we think actually we can very efficiently make a medicine. And so we don't have to take a huge amount of financial risk in order to do that, because the basic science has said this isn't as risky as we thought it would be. And so again, we've worked pretty broadly on rare diseases. The 5 that we've worked on are Fragile X syndrome, Fragile X associated terminal ataxia, ALS, dementia, Huntington's disease. I told you about Nancy. And what I'm going to tell you about today is work that we've done on myotonic dystrophy. And so myotonic dystrophy is the most common adult onset form of muscular dystrophy. And the diseases that we work on are ones that are caused, so this affects 1 in 8,000 people, and it's in families, it's genetically, you inherit this. And so one of the early things that I did when we first started our approach is we actually did one of our experiments, and the first RNA target that we came up with that bound a small molecule was actually an RNA target that bound a form of myotonic dystrophy. And so very quickly, I wanted to learn as much about this disease as I possibly could do. And so I went to a conference that the Myotonic Dystrophy Foundation ran. And it's a very interesting conference where what they do is they put in the same room the scientists and the patients. And so I remember, I remember to this day going into elevators with these patients, and there'd be a mom and there'd be a child, and very clearly they both had myotonic dystrophy. And so because of that experience and me learning about the disease, and the myotonic dystrophy community is both scientists and patients is a very special community, it really motivated me very aggressively to work for these patients to try to develop a scientific framework to get a disease. And so, you know, I've made it a point every time when I get into a new research area, especially in a rare disease, to go to a conference where the patients and the families are there, because you really get, you know, I'm an academic scientist and I work in a lab. As close as I get to these patients are growing their cells, right? I don't really get to see the families, and I try to really aggressively do that. And so we also did that in ALS. And you know, it's always motivated us to get the basic science done in as an effective and efficient way as possible to very quickly lay a foundation to accelerate drug development. And so the diseases that we work on are diseases that are mediated by what's called RNA repeat expansions. And so what an RNA repeat expansion is, it's like a broken record. And I do realize that our analogies are changing as we age. No person under probably 18 years old knows what a record is unless they buy vinyl because they think it's cool. But what happens is in these diseases caused by RNA repeat expansion is you get a massive repeat of 3 nucleotides. In the case of myotonic dystrophy, you get a repeat of CUG that's between 50 and 1,000 nucleotides of repeat. And what happens is this is a toxic RNA that folds in a way that causes disease in these patients. And so what we did is we used Inforna to design a small molecule that binds to this RNA target, which is shown here, that can cleave it. And so this is the model of this target that's rendered in 3 dimension that my lab, John Chen and Shruti Chowdhury, have studied in complex. And so how this molecule works is it binds selectively to this repeat expansion. It binds selectively to this toxic RNA, and it basically eliminates it, right? And so the consequence of it eliminating it in cells is it gets rid of about 50% of that gene. And it turns out that when you have these genetically defined diseases, you get 2 copies of the gene. 1 copy is a healthy copy, 1 is a diseased copy. And so we get about 50% of the total amount of the gene. It turns out we get about 90% of the diseased copy. So we're only eliminating the diseased copy of that gene. And so what I've shown here in this plot is that each one of these dots that are this dark gray are a gene that is dysregulated in the disease. And so if this thing isn't in the middle here and it's off the diagonal, it's basically affected in its function. And so what you can see that when we deliver this compound, and this is to a mouse model that has this disease, each one of those genes are improved. They go back to the middle here, where there's not a difference between this is a plot of the disease, this is healthy. And so it turns out by using this drug that eliminates the disease, we're improving about 97% of the RNA defects in this mouse model of disease. Now this is muscular dystrophy. And one of the ways in which muscular dystrophy manifests itself is you have an issue in contraction and relaxation of your muscles. And that's because you're missing a key protein that is expressed on your muscle fiber. And so when a patient has myotonic dystrophy, has the disease, a neurologist will ask them to squeeze a pen and they'll go release the pen. And it turns out they can't do that. It's very slow, whereas we can do this very quickly. And that's because these patients are missing a protein on their muscle fiber that controls contraction and relaxation. And the way your muscles fire is much like a battery distributes a charge. So you have to control gradients of ions to control that contraction and relaxation. And so in patients that have myotonic dystrophy, what happens is, and this is DM1, you don't see that green protein that's shown here. So this is what's called histology, or it's staining how much of this protein is in a muscle fiber. So in a healthy cell, you can see a lot of that green protein. Again, that controls contraction and relaxation. In a myotonic dystrophy, a muscular dystrophy mouse model, that protein is not there. However, when we treat it with this drug, you can see that protein reappears. And the consequence of that is that you affect and improve the ability of these mice to control contraction and relaxation. And so what I'm going to show you here are some images of mice that have been not treated with the drug and ones that have been treated. This is a mouse that has myotonic dystrophy. And these are videos by Amy Carpenter, who is a scientist at Expansion Therapeutics, which is a startup company that I founded. And so what's shown here is what you can see is if you go and pinch the hind leg of the mouse, you can see they have a cramp. So when you do that pinching, you can see the mouse is not able to relax its hind legs, and it keeps that cramp for a long time. That's what's called a myotonic discharge. That's basically the disease in the mouse where they can't control their muscle contraction and relaxation. Here's a mouse that we've treated with our drug that has the disease. So what you can see is as you pinch their hind legs, those mice are able to freely contract and relax their hind muscles. And so it's almost as if they don't have this gene. And so this molecule is able to eliminate the disease-driving repeat. And you can see that upon being challenged, that these mice don't have this contraction, that they're able to freely, they're moving around much faster in the cage. This pinch isn't having an adverse effect on them. So basically, not only at the level of their RNA defects have we improved the disease, we've upregulated a protein that's associated with contraction and relaxation in the mouse. And we've also seen an improvement in their ability to control contraction and relaxation. So I've been working on genetic disease since I started my lab 15 years ago. And you know, my ultimate goal here is to try to get a medicine for these patients that don't have a treatment option. Science is now in the news. And I wish that science would always be in the news in a good way. This is probably science in the news in probably not in a particularly good way. So this is coronavirus, right? So as many of you, all of you are aware, is that this is a virus that's being spread that initially started in Wuhan, China, that has now gone from all over Europe and all the rest of the world. We live in a world that is devoid really of borders. I have traveled to Japan many times, to China several times, and to Europe, and many of you have too. And so, you know, coronavirus just came, there was a pneumonia that was discovered on New Year's Eve of 2019. And very rapidly, we have over 190,000 confirmed cases with 7,519 total deaths that was updated yesterday by this coronavirus Center for System Science and Engineering at Johns Hopkins University. This is exploding and quite a significant issue that we all need to care for each other and not have this infection go by practicing social distancing, etc. Now, I want to make a comment about viruses. I get a flu shot every year because one time I got a flu really badly at Christmas and I stayed in bed at my sister's house for the whole week. And I was scared that I was going to give my nieces and nephews the flu. So every year since, I've gotten a flu shot. We know that every year there's a new strain of the flu. Every year we get a different vaccine for the flu. We know that viruses evolve, not only for the flu, but things like coronavirus. And so this is significant. 190,000 people as of yesterday are total confirmed cases. And this is confirmed cases. It's not probably the real number of cases. And so I want to tell you about viruses. So actually, the central dogma of viruses and most viruses, actually coronavirus, its genome is entirely built of RNA. So it doesn't have a DNA genome, it's an RNA genome. And so what happens is, because it has this genome, the way that viruses propagate, infect, and frankly survive is they integrate themselves into using our cells as a manufacturing factory to make more of the virus, right? And so viruses, again, have an RNA, their genome is RNA, and they integrate with our, protein synthesis machinery to make the protein, and they do it in a whole sort of ways. And so I'm going to tell you work that we've done in the lab very rapidly as a collaboration between Iowa State, with Walter Moss, Ryan Andrews, and Jake Peterson, and work that I've been doing in my lab at Scripps Research with Mason, Chris, John, and Hafiz. And so what we did is, the question we had is, and I'll give you some timelines here. The question we had is, could we very rapidly identify regions within the coronavirus genome that fold? And could we identify lead molecules that could bind to these targets? This is very much an evolving story, but we are always paying attention to health challenges and trying to deploy our resources to address it aggressively. And so coronavirus genome is 30,000 bases. So you can take 30,000 bases is huge. It's a 30,000 nucleotide RNA genome. Well, 30,000 bases is not so big when it's compared to the human genome that has 6.2 billion bases. And one of the consequences of this is that viruses, they have to use the host machinery. They don't have enough genetic material to bring along their own protein synthesis machinery. And so they just have an RNA genome that gets injected into a human cell, and then it hijacks our protein synthesis machinery to make more of the virus, to then have it replicate, kill cells, and go into the next virus. And so the viral genome is decoded by a host, or our ribosome. If we're infected, it's our ribosome that's decoding that genome. And it's very simple. There's very few segments in this genome. There's about 9 segments in the genome in total. 1A and 1B are 2 large proteins. S, E, and M are structural proteins that are the spike envelope membrane protein and nucleocapsid protein. They're basically proteins that allow the virus to form its structure. And then 3A, 6, 7A, 8A, 9B are translated into accessory proteins with unknown function. And so what we did is we looked at the structure of coronavirus and then identified about 525 RNA structured regions across that entire genome. And we identified 200 lead small molecule medicines that can bind throughout the genome. And so, for example, the virus has a region in it that basically causes a human ribosome, or the machinery that makes human proteins, to bind to it and make the viral protein. We know the RNA structure that causes that to happen. We've identified it, and we've identified molecules that can bind to our structure, to that structure. Those molecules in principle could bind to coronavirus structure and stop it from being decoded by the human host. So there's a lot of work that has to be done on this, but we have a lot of compound series that we're going through testing. And so I just want to give you a handle for the timelines here. This is going to really, look, it's good that we know this information. It's mission critical here. So if we wouldn't know and a lot of the testing that's being done is because we know the genome sequence of coronavirus, and people can do genetic, testing to see if the virus is there in very sensitive techniques. The fact that we know that the virus has infected 190,000 people tells us, hey, we have to take all the precautions necessary, such as social distancing and other things, washing your hands, and not transferring the virus to people that could be very sensitive to it. This is important to know information so that we can adapt to it. One illustration of that is that the South Koreans had a very robust testing regime, and because of testing, social distancing, and changing normal behavior, the virus was petering out. The rate of its growth was greatly slowed, and I hope that the U.S. is able to mirror some of that. We're getting ready now. So I want to show you some of the timelines here. And so, literally on New Year's Eve, pneumonia was detected in Wuhan, China, and that was reported to the World Health Organization. In January 7th, 7 days later, or 8 days later, the virus was confirmed as a novel coronavirus. In January 12th, that genome sequence was published. So 5 days after it was identified as a coronavirus, people were able to grow it in a lab and sequence its genome. It's absolutely amazing. We got involved in February 1st, where we started to collaborate with Walter Moss's group, Ryan and Andrew, to identify regions in that genome structure that were highly structured. And we identified within 2 days about 524 stable structure. At February 11th, this was called COVID-19, was the virus's name. And in February 17th, we identified about 200 lead compounds that are targeting the viral RNA, and we're ongoing testing these molecules. So our goal here is to try to identify molecules that can stop the virus from hijacking our protein translational machinery and stop the virus from getting packaged, by getting synthesized, by interacting and interfacing in the propagation of this virus. But these are things that man has dealt with throughout the ages. And so we just have to be able to use this. Now, there are many groups that have been mobilized to target coronavirus. There's people that are making antibodies, or they're making a vaccine associated with it. Mike Farzan at Scripps and Cho, and people like Christian Anderson, Andrew Ward, Ian Wilson are working aggressively on this problem to make a vaccine. That's if you can vaccinate before you get the disease. People like my lab, Caliber, Pete Schultz, Arnab Chatterjee, they're doing high throughput screens to identify chemical matter that can affect the virus after it's infected you. Both of those approaches are very important because you can use a vaccine before you get the virus. Once you get the virus, the vaccine is not necessarily going to be effective. And so you really need to give medicines that can stop the short circuit the viral from being infected. But all of this work is at an early stage. It's going to take us a year or so at least to get a vaccine. It's going to take us that long to finish clinical trials to get a medicine. And so I think one of the really, I think, awesome things that Scripps enables us to do is we have Caliber as our translational arm. And so the basic science that we do here, the foundation that we've built, allows us to rapidly deploy those resources to emerging threats that come out. And it's going to be inevitable. Next year, there's going to be a new flu. It could be a different coronavirus. And so we need to be able to build our scientific infrastructure to react to this very quickly. And so these are challenging problems that we need to address. And now everything that I talked to you about is just the beginning of making a medicine. And so what I started this talk on was rare disease. And then I ended on something that hopefully stays relatively rare, which is coronavirus. And so what you do in drug design is you design a drug, make a drug, test it in cells, test it in animals, and you make sure it's safe. The goal, and what you don't want to do, is you don't want to deliver a medicine to a patient that's not safe, right? You want to make sure that these molecules are both safe and effective, and that's why the FDA plays critical roles in these pathways. And so key questions that one needs to think about when they go to make a medicine is, are there enough patients? Do you have the technology to measure drug activity? For coronavirus, we have the technology. You can detect if the virus is replicating and what's the amount of load. For things like muscular dystrophy, you can detect if you're improving the muscle strength and muscle contraction and relaxation in the patient population. For cancer, you can see if your tumor has diminished. And you also need to understand what's the safety profile that you need for a drug that's going to treat a disease. If you have a very serious case of cancer, maybe the drug doesn't need to be as safe because of the urgency of the medical need than you would for muscular dystrophy, that a patient's going to take this forever. And so these are very challenging, complicated biomedicine problems that really need to be addressed, I think, by merging basic science with people that are developing medicines, which we're blessed to be able to do with our partnership with Caliber, but also biotechnology infrastructure broadly. They need the basic science that people like Scripps develop, and they need us in many, many, many ways. And so I want to sort of end you here is that 1 in 10 humans has a rare disease, and you need many rare people to solve the problem. I started a biotechnology company about 3 years ago, where the goal is to make medicines for patients that have these RNA-mediated repeat expansion diseases across the earth. This is what we call rare disease. Myotonic dystrophy is a rare disease, but there's about 1 million people across the earth that have this disease. So that's not rare when you think about 1 million people. And the NIH has a calculation where actually on aggregate or together, 1 in 10 humans has a rare disease. So even rare diseases are not rare, right? These are very important medicine. And so not only do we need rare people that are CEOs of companies, like Kevin Forrest is the CEO of our company, Suzanne Zuchek and Sai Velagapudi. This is Amy Carpenter, who gave us did those videos, Elliot Eric, Scott Rockledge, who is a venture partner at 5AM, who was the funder of Expansion, Tim Blizzard, who directs our medicinal chemistry effort. You really have to work in partnership with these people. And so we've been blessed in many ways to be able to do that. And I've been blessed to work not only with those people, but the people in my lab. I think at the end of the day, all of business is about people, especially when you're in an academic research center, where we do really hard things here. But the output in those hard things hopefully is worth it, because, you know, at the end, you know, we might not be the people that cross the finish line, that gets, you know, breaks the tape to get the drug, you know, when you finish the marathon that is making a therapeutic. But, you know, to contribute a few miles in that race is really what we want to do. And so, you know, we're thankful to be able to do that. And so, yeah, that's all I had to say. And I'm happy to take questions here. So if you guys want to answer questions, I can answer questions. Right. So, okay, so Sebastian Brunenmeyer is asking a question. So he goes, what are the advantages of small molecule RNA binders over siRNAs and ASOs? And so why are there so few approved RNAi therapeutics? And so my lab is working to make small molecules that target RNA structure. Another way to target an RNA sequence is to use a designer piece of DNA that binds to the RNA. So these are very high molecular weight compounds that you can't take by a pill. These are molecules that you'd have to inject. And so the advantages of small molecule RNA binders is you don't have to inject them. You can take them as a pill. And so the consequences of that is there was recently, probably 3 years ago or 2 years ago, an antisense oligonucleotide that was approved to treat children that have spinal muscular atrophy. So this is a disease where these children don't have neurons that are developed because they have a genetic mutation. And so this is a drug that is related to the world's most expensive drug. So this is a molecule that has to be injected directly into the spine of these children. And the drug regenerates their neurons in their spine, but you have to constantly be injected into your spine. And that's not an easy thing to take. In fact, if you have to deliver medicines by injection into the spine, you might not get compliance from patients. It's not as easy as taking a pill. The other issue with antisense oligonucleotides, which is related to that, is they don't have broad tissue distribution. And so actually there's been a huge amount of progress in identifying what are the types of medicines that antisense and siRNA should tackle. And so that Alnylam, which is the medicine company that's using siRNAs, has actually had a large number of approvals for going after the liver, which is where they can engineer these molecules to get to. The other issue is that, you know, developing therapeutics takes a lot of time. Even though Biogen, Ionis, and Alnylam have been at it for a while, now that pipeline's starting to accelerate. One, for liver diseases, and two, for diseases that are associated with the brain, because you can intrathecally deliver it and it'll get it to the brain. The second question that Sebastian is answered is, how many contact sites do your compounds use to interface with RNAs? Are these molecules also DNA intercalators, and how do you assess selectivity of binding in whole cells? Yeah, this guy's a scientist. So these molecules will bind selectively to RNA. They often, it depends on the compound, the number of nucleotides that they interface with. It can be up to, you know, 10 nucleotides of three-dimensional space. And we assess selectivity by basically figuring out what RNAs these compounds bind. We develop technology to do that broadly. So Sebastian, we have a Journal of the American Chemical Society paper in 2019 that'll tell you all about that. Jay Streeter asked, how many of the potential how many of the potential 200 COVID-19 compounds are already approved medicines? So there's about 10 of them that are already FDA approved medicines that we're aggressively putting after. Now, the reason you're asking that is that if we're repurposing approved medicines, basically the approval timeline would be greatly shortened. And so we're very interested in those compounds. Actually, that approach is the one that Caliber has taken, because Caliber is rapidly screening medicines against COVID-19 that are FDA approved in collaboration with the Gates Foundation. And so their goal is to have molecules that can very quickly be put into patients because they know that they're safe in them. Rhonda Shea is asking, how close is your work to doing a trial for a medicine to treat DM1? And so actually DM1 is why we founded Expansion. And so, you know, we could be years away from that treatment. We're just, we're advancing compounds through preclinical testing. And so we're optimistic that we will be able to do this sooner rather than later, but these are very difficult problems to solve. And so we could be realistically years after, years from developing a therapeutic, but we hope that sooner rather than later. These are very complicated. Nancy is asking, how long does the small molecule treatment last? How often do you need to take more? So that depends on the disease. So if it's coronavirus and we can eliminate the virus, or it's a flu, once we deliver that drug and we eliminate the viral infection from a patient, they won't have to take that medicine anymore. If it's a cancer, you know, you'd ideally like to be able to take a person to take that every day. Once they get rid of the cancer, they wouldn't take that medicine anymore. Nancy's also asking, can you comment on Moderna's mRNA vaccine for coronavirus? So Moderna is a company that is delivering medicine, where their medicine is deliver a whole messenger RNA. And so what they're doing is they're delivering to patients a messenger RNA that can produce a virus, a messenger RNA that can produce a vaccine in patients. And so that's very early. It's very exciting technology because Moderna was able to very rapidly identify a lead for making a vaccine. Actually, one of my science heroes is the CSO of Moderna, Melissa Moore. And if Melissa's working on it, it's going to happen sooner rather than later, I have no doubt. And so they just started delivering this vaccine to patients. And so what they have to do is they have to determine if patients produce the vaccine with that messenger RNA first, which they're trying, if it's safe. Second is, are the patients producing that vaccine? And third, is that vaccine efficacious? And so this is going to take a while. I mean, it's just going to take a year. Okay, Rhonda is asking, in the case of multiple organ systems being affected by myotonic dystrophy, would a therapeutic target all systems at once, or would you separate medications, or separate medications be needed for each organ system? Actually, that's one of the major advantages, or one of the major potential advantages of small molecule medicines. So myotonic dystrophy, although I articulated that it's a disease due to muscle contraction and relaxation, it's many organs that are affected in this disease. And so the organs can be your heart, your diaphragm, your skeletal muscle, and it can also be your brain, because many of these patients are affected by issues of sleep. This is a multi-system disorder. And so one of the advantages of small molecule medicines, in contrast to these oligonucleotides that Oscar, I believe, asked earlier, or Sebastian asked earlier, is that a small molecule medicine in a pill, we could be able to engineer that to get into your brain, to get into your heart, to get into your diaphragm, and to get into your muscle, whereas an antisense oligonucleotide is not going to be able to do that. You're going to have to inject it to deliver to the skeletal muscle. And then if you want to treat the CNS issues, you'd have to inject it into your spinal column. And so that's one of the advantages that our approach would have for small molecules, but we have to deliver on that promise. And we're not quite there yet, but we're hoping we can do that. Oscar is asking, can you comment more on the RNA motifs found on the coronavirus system? So sure, we have RNA folds throughout that system, that we have drugs that bind to it that can affect integration of that virus with the host, that can stop its translation, et cetera. And so it's a lot of complicated science that we have within these folds. We know within coronavirus folds that are unique within that virus and generally not thought to be present in the human genome that could be more selective, and we're working through that. Right, Larry Greenfield says there's a known drug that treats rheumatoid arthritis, and now there's considerable consideration to use this to treat the new coronavirus, and can this be treated as an orphan drug approval? So there are expedited ways to get known drugs to treat patients. There's a lot of reports that are coming out that approve medicines for other indications are being efficacious against coronavirus. These are early days, right? You have to make sure that these medications, even if they're repurposed, are doing no harm to these patients that have the disease. And so, sure, these molecules could have an expedited approval to treat coronavirus, but making a medicine is a very complicated issue. Given that it's a known medicine, there's a high possibility or high probability that it'd be safe to a patient, but we don't know if that medicine would be safe for someone that's infected with coronavirus. And these are the types of things that we have to be addressed in a clinical trial. Shinsuke Segawa is asking, can RNA binding compound degrade the RNA or reversibly bind them? So our molecules bind an RNA, but we also can degrade them, and we can degrade RNA targets with small molecules in two ways that the lab has published. One way is we can directly cleave them, and by basically appending these compounds, it's what's called natural products. So these are molecules that are produced in nature, bacteria make them, we can append them to RNA targets and they'll directly degrade the target. The other is we've made an approach where we can actually recruit. So cells make enzymes that degrade RNA, right, very rapidly. And so we've developed an approach where we can make small molecules that bind to an RNA and bring those degradation machineries to cleave this RNA in a very efficient and effective way. So we've been able to do this very broadly. Yeah, so Curtis is asking, speaking of drug repurposing, do you screen approved known compound small molecule drug against your RNA libraries? Curtis, actually, it's a very interesting question. One of the issues that we had early on is people didn't believe that known medicines would bind RNA. So in collaboration with Caliber, we've taken their reframe library. So it's a collection of 15,000 molecules that have all been put into man. And some of them are approved drugs. And we've actually shown that those compounds broadly bind to RNA targets. It's interesting, about 2 years ago, we were asked by someone at a local event we went to if we were interested in targeting Alport syndrome. I had no idea what it was. And so I looked at some of the work that we had done in defining known drugs that could target something that would be associated with disease. And it looks like we have a molecule that could be useful for that disease. And so yes, broadly, we've been able to repurpose drugs that are traditionally protein targeted medicines to bind RNA. And what's also been interesting is that, when many drug companies go to advance medicines that target protein, they oftentimes don't consider RNA as an off target. So not only have we been able to repurpose medicines for binding to RNA broadly, we've been able to demonstrate that in many cases, when people go to show that medicines do no harm, they need to see if these medicines not just affect protein, but also affect RNA. George is asking, can you tether the gene block for replication so repeat injections wouldn't be needed? Affix the block and force the system to repeat the copy. So actually, for some diseases like myotonic dystrophy type 2, we've actually developed an approach where this small molecule drug will actually trigger a disease cell to eliminate itself. We've actually published that. And so this is very interesting. So we can actually target these genetic diseases and force the gene to get processed in a way that it eliminates itself with a small molecule medicine. For the scientists in the audience, we published this for myotonic dystrophy type 2 and cell chemistry and biology in the past week. This guy, Raphael, he did that work, who's a student, a postdoc from Israel that's joined us, really done amazing work on this. And the thought is that many of these molecules wouldn't have to be given a repeated delivery, but that's really needs to be investigated on a case-by-case basis, because it could be repeated delivery could be a function of how long the medicine sits in your body, and also what is the effect that the medicine has on a target. If the medicine affects the target by simply binding to it, then you might need to more often administrate that. But if the medicine works by eliminating it, you can imagine a situation where you have to give this drug more frequently. Fior Breslau is asking, is there any new drug for ALS? Actually, about half of my lab is devoted to target developing precision medicines for ALS. We actually have an NIH program project grant that I'm the director of in collaboration with Jeff Rothstein, who runs the Packard Center at Johns Hopkins University, and Len Petroselli, who works at Mayo Clinic in Jacksonville. And so that program project grant is devoted to laying a foundation for medicines where we are developing and delivering small molecules that target medicines associated with the most common form of ALS. My lab is also working pretty aggressively to develop medicines that deal broadly with some of the damage, such as what's called neurological inflammation that's associated with not only ALS, but dementia and Alzheimer's, and we're working pretty aggressively for that. I think ALS is probably, I just want to opine on this. So, my mom had Alzheimer's, and as many of you are aware, many of our fellow citizens are affected with Alzheimer's disease, and yet the drug companies have had a lot of failure. There's not a lot of success that's happened in Alzheimer's, in part because it's very difficult. You don't know when to treat these patients. You don't know how long you have to give them a medicine for. There's a lot of missing stuff in the basic science. What we do have is a lot of very good biomarkers. So we know in principle if our medicines are working. One of the, I think, advantages, and I don't want to be sort of glib here in that, is one of the things that helps ALS drug development is that these patients, their decline isn't as gradual as, although, my mom had I think anytime your mom has Alzheimer's, you don't think it's gradual, you think it's pretty rapid, frankly, right? But whatever rate of decline she had, it's nowhere near what happens in an ALS patient. And so because their disease manifests itself very quickly, there could be a way that a drug company could rationalize intervention to see if that progression is slowed in a very, the timelines for doing that clinical trial wouldn't be as long as in Alzheimer's. And so, we're working very hard. We have, Thomas is also asking a similar question, is, in the medicines you may be creating for ALS, as you identified the defective RNA is causing the death of the motor neurons? If so, are the medicines designed to stop the progression, destroy the motor neurons, or both? So our medicine is designed to basically edit this gene out of patients. And so we have actually seen in some preliminary studies that we've eliminated the gene and that there has been some repair in a mouse. But if that works in a patient, who knows? It's very early days, but we're working very hard to answer all of these important questions. I mean, our hope, our prayer, frankly, is that it would repair something, but it's just a prayer. More realistically is that these molecules would slow the progression. But we just don't know for a lot of these neurological diseases if the brain is capable of broadly repairing itself, or we would just hope to slow the progression. And that's, those are some very fundamental, basic scientific questions that we need to understand for this. I'm just making sure I've answered all the questions. D. Tesler is asking, can new RNA tech help to understand why known drugs like chloroquine for malaria appear to be effective on COVID-19? Yeah, it can. So we actually have technology that I didn't describe here that can tell us if medicines bind to RNAs in COVID-19 and where they bind. And understanding how these molecules interface with the virus, if they bind a viral RNA, if they bind a protein, and where they bind, can help us understand how these compounds work. It's not only important to make a new version of chloroquine, but it's also important to understand if the virus becomes resistant to those medicines when they're deployed, how to make a new compound that can attack that resistance mutant. And so, yeah, the technology that we're deploying and we've developed in the lab can allow us to understand what are sites on both RNA and protein that these compounds are binding, to be able to understand how the molecules work, which can help us understand the basic science of how to affect a virus broadly, but also how resistance mutations can emerge to get hopefully one step ahead of resistance mutation. George Kohlberg is asking, will peptides bind to RNA folds broadly? So yeah, there are peptides can bind to RNA and they can bind to RNA broadly. People have worked on getting peptides to bind to RNA. There's a challenge with making peptides as medicine. because many peptides, if they're built from natural peptides, are subjected to decay. So people have worked on that. Picardi R is asking, since it seems that coronavirus activates IL-6 or interleukin-6, part of our immune response, that one of the targets of RA drugs, do you think that studying coronavirus RNA would be possible to study IL-6 activation in rheumatoid arthritis? Maybe, I don't know. So I think you're talking about, yeah, there's a lot of activity within repurposing medicines that are studying over immune response that have been applied for basically issues of over immune response, such as rheumatoid arthritis, and repopulating them for coronavirus, and that's an active area of research. Ines is asking me, have you ever considered working on drugging RNA in genetically inherited pancreatic cancer in a similar way to triple negative breast cancer? So yeah, we're actually working on all cancers broadly. One of the areas that we're working on is inherited pancreatic cancer, where there are some genetic associations that we think we can target with RNA associated small molecules. Carl is asking, I'm curious to hear if you tend to find similar small molecules that interact with RNA, and if you see any trends that could be included in future libraries. So yes, actually we found that many FDA approved medicines that target kinases actually target RNA broadly, and there's overlap in approved medicines and the structures and folds that they have associated with the structures and fold that medicines have that bind to RNA. So we found that. And that has actually helped us to develop new libraries of compounds that can bind RNA more avidly. Sebastian is asking, might viruses like coronavirus mutate resistance to RNA binders as rapidly as protein binders? They might, but keeping in mind that what we looked at for these RNA structures are what's called evolutionarily conserved structures within the RNA. And so in order for that virus to mutate to an RNA structure, it only has 4 bases again. It has to mutate, and it has to mutate in a way that the RNA doesn't bind a drug, but that RNA fold still functions in a proper way to continue to have the virus to be propagating. Some would think that it's harder to mutate around molecules that target RNA than protein, but that remains to be seen. One of the things that I like about Inforna is that this computational approach allows us to identify a mutant of COVID-19, figure out how that mutation affects the RNA fold, and then make another molecule very rapidly that can affect that mutation. Now, this is a thought exercise, but it's something that I think we're really excited to deploy in these systems to tackle molecular recognition issues. Right, Herb Sarnoff is asking, are there ways to target your technology towards upregulating genes for haploinsufficiency driven conditions? Yes, so that's one thing that we've been very interested in deploying. And so for example, there are many diseases where, what's a disease of haploinsufficiency? You have 2 copies of your genome, and one of these genes is mutated. And so you have one that's healthy. So the question is, how do you boost the healthy version of that gene to make up for not having the second copy? And so the way that you boost that healthy gene is there are many non-coding RNAs that silence this healthy gene that we know that you can boost them. And so we've actually been able to do that for some genetically defined autisms broadly. And we think this is a very exciting way in which RNA targeted small molecule medicines could interface in making new approaches. Yeah, so I think I've answered most of the questions today. I want to thank you all for taking the time to listen to me talk.